So here we have mitosis, right? So what's the first phase of mitosis? So the first thing is like before mitosis, right? So it's
Oh, that's what we're talking about?
it's interphase, right? Interphase. So what happens throughout interphase? What's like the most important thing? So we replicate our DNA, and then what are those other two stages? So the two big things there are we replicate DNA and grow cell. Very good. All right. So what is the first official phase of mitosis? Prophase. All right. So what happens in prophase? So the DNA compacts. Yeah. What else? Yeah. So yeah, the mitotic spindles form. And where do the mitotic spindles come from? Yeah, this. So yeah, the centrosomes, which are made of two centrioles, right? So come from from centrosomes. All right. So then, what's the next phase? Prometaphase. All right, and what happens in prometaphase? Yeah, the nuclear envelope disintegrates. What else?
Is this when the sister chromatids like come together, or sister chromatids are formed?
Well,
That's what they attach to.
well, you can kind of spin that into but that. So we replicate our DNA in the S phase, right? So what happens to the DNA itself from here to here?
It attaches to the little spindles.
Okay, yeah, that's another thing that happens, right? Chromosome. Well, before that happens, even.
It's duplicated, right?
Yeah, it's duplicated where? In the S phase. Right. So
Wait, what'd you ask?
what happens to the DNA itself before we attach it to the spindles? Kind of. So it compacts more, right? So it goes into this remember this picture that we have it looking like this, and then it compacts more into this phase. So it's got to compact more, and then when it compacts more, that's when the kinetochores form, right? And then after the kinetochores form, then we do whoever said that step of attaching to the spindles, right? So nuclear envelope disintegrates. So the DNA compacts even more into chromosomes. Your kinetochore appears. And spindles attach. Very good. All right, so what's the next phase? Metaphase. So what happens in metaphase? So the chromosomes align on metaphase plate. Right, and that's about it, right? That's all the important stuff. So what's the next phase? Anaphase. So what happens in anaphase? So what about the chromosomes are pulled apart? The sister chromatids are separated, right? So the sister chromatids are separated into distinct chromosomes, right? And then what's the next phase? So the next phase the next two are kind of combined oftentimes, but we're going to separate them here. So what's right after anaphase? Telophase, right? So telophase. So what happens in telophase? So the nucleus reforms. Good.
And the ones that aren't attached to the chromosomes expand.
Well, actually, that happens in anaphase. So yeah, we didn't put that, but the non-kinetochore microtubules lengthen. All right, so that happens in anaphase, but good job for remembering it. So the nucleus reforms, spindle fibers, but what happens to the spindle fibers? They go away, right? Disappear. And that's about it. And that's it, right? So the last one is,
What about the cleavage phase?
so yeah, we'll put that there. So cleavage. Actually, no, that doesn't go there. Sorry. Where does that go? Cytokinesis. So cytokinesis. So there's kind of two things that happens in cytokinesis. One of which is cleavage furrow forms, but another thing before that. It's kind of the most important part of cytokinesis.
The microfilaments right on the
No, not quite. Something with the cytoplasm. Yeah, so we replicate the cytoplasm. Because if we have a cell, right, and it has like one Golgi apparatus, and then we're going to replicate it, that Golgi apparatus is either going to go in either cell. So we have to make sure we replicate that Golgi apparatus so we can put a Golgi apparatus into each new cell. Right? So we have to replicate cytoplasm. And that's what cytokinesis is. Like cyto is the cytoplasm, right? And then kinesis means movement, right? But it's like movement of the cytoplasm, like replication. But yeah, so replicate cytoplasm and cleavage furrow forms. Yeah, very good. So that's just a review of mitosis real quick, which is going to be very important for this session. So, how many chromosomes are in each body cell? Just 46. Very good. All right. So we're going to talk about in this session primarily is the human life cycle and meiosis. Okay. So what we're going to do is we're going to go through the human life cycle. But this applies to all. Should I put this in that conference? I'll just. That's right. All right. So here, so we know that each cell has 46 chromosomes, but that's like a cell that functions. So when we do asexual reproduction, we have to fuse a sperm and egg, right? How much DNA needs to be in each? How many chromosomes need to be in each cell that we fuse? 23, right? So we have to. So the process of meiosis divides that in half. Okay. So what we have is we have a male and female, right? So we all know that females have ovaries with eggs. And males have testes with sperm. Right. All right, and so the way that we create those is the process of what? Meiosis. Yeah. All right, and so we're going to create these two things, and then we're going to fuse. So when you go through meiosis, you create two cells, like these two. Both of these cells have 23 chromosomes, like we said. What do we call a cell that has 23 chromosomes? Haploid. Yeah. Okay. So both these are haploid cells. All right. All right, and then these two cells right here are, so these are your sex cells, right? So they're your cells that have to do with reproduction. They have the scientific name. Does anyone know what they're called?
Gametes.
Your gametes, yeah. So your egg and your sperm are your two gametic cells. Right. So then, when we fuse these two things, it's the process of what? Fertilization, right? So fertilization. All right. And so when we fertilize an egg with a sperm, we get this cell right here, which is called a zygote. It's one of the best words in Hangman. All right, just Z-Y-G-O-T-E. Like no one guesses those letters. So you can use that one. That's free. That one's free right there. Anyway, so you have the zygote. All right, and then these zygotes can undergo. So this is a fertilized egg, but it's a singular egg. So if we're going to create a human from this fertilized egg, what process do we have to go under to make that human? Mitosis, right? So when you reproduce a cell that has the proper number of genetic material, you have to go through mitosis, all right? So this is mitosis into an adult. Makes sense? Anyone have any questions about any of that? Tyler's not saying no, so. Kyle. Anyway, okay, so somatic cells, right, are, does anyone know?
That's like stem cells.
Yeah, it's your body cells, right? So it's your non-sex cells. So body cells are somatic cells. Your gametes, like y'all said, are your sex cells involved in reproduction. Sexual reproduction is, so it's the reproduction that's involved with two separate, like partners, right? So it's the combination of genetic material from different individuals. And they have to be like opposite sex and all that. Right? It's kind of, and then asexual reproduction is just reproduction of an organism to make identical cells. And whether or not they differentiate is up to the organism, right? So zygote is, we said, a fertilized egg. So if you're diploid, you have how much? Or what are you if you're diploid? So yeah, you have both sets of what? What kind of chromosomes? What kind of chromosomes? Homologous chromosomes, right? Homologous chromosomes. While if you're haploid, you just have, one set of all right, and then an autosome is it's your chromosomes that determine No. Oh wait. Okay.
Not just non-sex chromosomes. Right?
Yeah, it's just non-sex chromosomes that do not determine sex, and then your sex chromosomes are. So a lot of these words like make sense, right? Some of these like you can maybe not exposed to a lot of times, but like when you think of like asexual reproduction, like you can kind of intuitively figure that one out. All right. So like we said, so how meiosis and mitosis are involved is they are distinct processes, right? Just one is involved with creating what? So what is meiosis involved with? We'll do that one first. So yeah, two different haploid sex cells, and one is involved with just replicating an organism with the same number of chromosomes, right? So they're distinct processes. Right, they are distinct processes that are they distinct processes. Mitosis creates. So mitosis creates identical cells.
Doesn't meiosis create 4 haploid cells?
Yeah, it does. If you want to do the number, you can do that. This one creates 2 identical cells. Right. So that's another difference. But we're going to talk about the differences between meiosis and mitosis later. Before we do that, we're going to talk about crossing over. So, does anyone know where crossing over happens? Prophase of metaphase of meiosis 1. Right. So what we're going to do is we're going to just talk about the general process of meiosis. So it's all regulated by enzymes, like most biological phenomenon are. So what happens is your chromosomes are going to line up, right? And so this lining up right here is called a synapsis. Right. So the lining up of homologous chromosomes in prophase 1 is called synapsis. So the lining up right. So after they're lined up, they're going to cross over there. They're going to 2 non-sister chromatids right here are going to cross over each other and like touch each other pretty much. All right. So this interaction of the 2 non-sister chromatids is called a chiasmata. All right. Interaction of 2 non-sister chromatids.
Isn't there a name for like where each code is on the DNA, like locus?
Yeah, the locus.
Is that right? Is that what it's called?
Yeah, it's a loci or a locus. Yeah. So there's going to be a lot of enzymes that like you're going to cut your DNA and like replace it, and it's all there's a lot of enzymes here. But what you get is after you cross over and you exchange this DNA with each other. Okay. So if you have DNA in one place that came from another place, it's called recombinant DNA. So DNA that is exchanged with another source of DNA. So this isn't the only place that we see DNA and recombinant DNA in biology. Right now, like I'm in this biochemistry lab class, and what we do is we take DNA that we like completely made ourselves, and we put it inside a bacteria, and we made the organism express that that DNA that we made, and putting the DNA inside of that bacteria makes a recombinant DNA. So anytime you have DNA from another location and you put it into another place, it's called recombinant DNA, and it pops up a lot in just some weird places every now and then when you're like reading like articles and stuff. So it's a general term. It doesn't just happen here, but this is a very good example of it. And then your non-sister chromatids right here are in crossing over. The non-sister chromatids are what exchange DNA, and they're on homologous chromosomes. So in crossing over, they exchange DNA between homologous chromosomes. All right, so does anybody have any questions about any of that vocab or any of the processes between crossing over or the human sexual life cycle? Beachy. All right. So the next thing we're going to talk about is just like we did with mitosis, we're going to go over meiosis in its kind of detailed stage. Okay. And it looks a little bit more complicated because there's a couple more steps, but it's actually not bad at all, right? So before, I actually don't have a picture of it, but before, so just like in mitosis, before we start this whole entire process, we're going to go through G1, S, and G2 phase. So we are going to replicate the DNA. So we will have 2 sister chromatids on the chromosome, right? So before we do this whole entire process, we replicate the DNA. So what happens is the first one. So the steps exactly mirror mitosis. Except for, with one exception, is there's no pro-metaphase. There kind of is. I don't get why they don't just label it as a thing, because like the steps still occur, just they don't actually label the steps. They put some of the steps in prophase and some of the steps in metaphase, so they make it distinct, but there's no technical pro-metaphase in meiosis. But everything else is the same. Okay, so what's the first step that we're going to go through? Prophase. So prophase, right? The only thing is each step, prophase, metaphase, anaphase, and telophase, they're going to happen twice, so we have to label that. Okay, so it's prophase 1 when you do it the first time. All right. So we said earlier that in prophase 1, you have crossing over occurring, right? But just like in, so what happened in prophase of meiosis? Or I mean mitosis, sorry. Yeah, so we start compacting our DNA. All right, and then what else happened? Look at that purple stuff. Yeah, your nuclear envelope dissolves. And then what about the yellow stuff? So what are these organelles called? Centrosomes. So your centrosomes, produce microtubules, and those microtubules are called spindle fibers. All right. So the next phase is prophase 1. So what's the next phase? So metaphase 1. So what's going to happen in metaphase 1? So be a little bit more specific than chromosomes. What kind of chromosome? Yeah, so homologous pair of chromosomes line up. So that's a little bit different in mitosis, because what lined up in mitosis? The sister, so just the chromosome itself with the two sister chromatids, right? So also in metaphase, what happened? Well, where is that happening? Right, so synapsis happens here, right? Because you have to, before we start crossing over, we have to line them up, right? So they're not lined up along the plate, but they are like lined up with each other. Does that make sense? So you see how these aren't on any plate, but they are like paired up with each other. The homologous chromosomes are paired up. So synapsis happens in prophase. But in metaphase, what's going on with maybe the yellow things? So yeah, the microtubules attach to kinetochore. And then there's that one, what's that one last step that happens in metaphase? Oh, wait, we said it happened in pro-metaphase before, but in mitosis we said it happened in pro-metaphase, but what's happening with the DNA? Oh, what? Yeah, it condenses more. So further condensation. Okay. So that's metaphase. Then what's the next phase? Anaphase 1. So what happens in anaphase 1? There's kind of two things. Yeah, so separation of homologous pairs and just the last little side thing with the microtubules. So yeah, the non-kinetochore microtubules lengthen. Y'all remember what the difference between a kinetochore and a centromere is, right? So what's the difference between the two?
Doesn't most things have to do with their function?
Yeah, it does have a lot to do with their function.
But doesn't centromere hold the two together and the kinetochore are attached?
The two what together? Yeah, the two. So the centromere hold the two sister chromatids together, while what does the kinetochore do? Yeah, so that's the location where the spindle attaches. All right. So the kinetochore is it's an additional protein that is attached to the centromere. Okay. But one is DNA. So the centromere is a region of DNA that connects the two, but the kinetochore is a protein attached to the centromere that also attaches to the microtubule. But very good. All right. So next is what? Telophase 1. All right.
Move that down a little bit.
Yeah, sorry. So we got telophase 1. So what happens in telophase 1? Yeah, isn't that kind of weird?
Yeah.
That we're reforming a nucleus just so we can dissolve it in a couple seconds, right? But it actually happens.
Is that the same thing with cytoplasm?
What?
It's the same thing with the cytoplasm?
Yeah, so you will have cytokinesis still. And you replicate cytoplasm. One thing that's important to note. So these two things, with crossing over, you see how we cross over, right? But we're still, and then what happens is we cross over and then we exchange the DNA, but you're still crossing over. When you go to pull apart the two homologous chromosomes, right here in anaphase 1, when you start to pull those apart, the DNA is still overlapping. Even like it's after the exchange of DNA, but it's still overlapping like that. But when you pull it apart, that's when it's like becomes unoverlapped. It's a technicality, but there are. I had a test question on my test when I took level 1 about that. But I don't know if like y'all's teacher will do it or not.
Like, how would you word that in a question?
Yeah.
So they overlap until
I forgot. I just remember I got it wrong. What?
They overlap until anaphase?
Yeah, so they overlap until anaphase, and then when they're pulled apart, you can kind of see in the picture too, right? Like if you look closely, you kind of see how like they're still overlapped here, and then even though they're fully exchanged. But yeah, so just note that.
So you say they're overlapped until anaphase?
Until they're pulled apart, yeah, in anaphase. All right, so then we go to telophase 1. So now we have two distinct cells, right? That each have one of the homologous pair. So, what's going to be this first phase?
Prophase 1 or 2?
Prophase 2, yeah. So we got prophase 2, and then what happens here? So same general stuff, right? One last thing is that the DNA relaxes. The DNA relaxes in telophase 1. All right. So what we have to do is, so what's some stuff that happens?
The DNA compacts here.
So DNA compacts. What else? The nucleus dissolves. Centrosomes produce mitotic spindles. Right. So same exact, like same general stuff, right? It's just a different amount of DNA.
Does
Yeah.
the nucleus and the nuclear envelope dissolve?
So the nucleus is the nuclear. Okay, I guess you're right. So the nuclear envelope. So the nucleus is the nuclear envelope and the DNA inside. The nuclear envelope is just the lipid around it. So the nucleus is the region in the cell that has a membrane and then the DNA inside of it. Like it's a region in the cell. While the nuclear membrane is just the outside of it, right? It doesn't have the DNA inside. So yeah, technically the nuclear envelope dissolves. All right. Anyway, so what's the next phase? So metaphase 2. All right. So what happens here? Yeah, the sister. Where else have we seen sister chromatids lining up? Yeah, mitosis, right? All right. And then what's that other step in metaphase 2? Yeah, the DNA further compacts. But what happens with the microtubules?
They're attached to another phase.
To kinetochore. Alright, so microtubules. Was that your question? No. So yeah, your microtubules attached to your kinetochore. Did you have a question?
Yeah, like with the prophase too, they're like, so that just only occur once, like in prophase 1?
Yeah, so it doesn't occur in prophase 2 because we don't have a homologous chromosome to overlap with, right? We separated those. A good question, yeah. So yeah, that only occurs in prophase 1. Oh, shit. So I forgot one more thing in metaphase. A lot of stuff happens in metaphase 1, right? One more thing in metaphase 1 is yeah, the asters form too, but that's kind of a technicality. But it's technically okay. Asters, right? Asters form. But you have what's called an independent assortment. All right. So does anyone know what independent assortment is? All right. What independent assortment is that so we have these two. So you have homologous chromosomes. You have one from your dad and one from your mom, right? So you can put your dad in red and your mom in blue. Right? So your chromosomes don't line up like all of your dad on one side, all of your mom, all the chromosomes from your mom on the other. What happens is that they could like mix and match. And that's what's kind of going on here is that you have your mom here and then dad, and then the cell will get your dad here, your mom here, right? And that these things assort on the metaphase plate independently. They don't like all coordinate. Like getting like all your mom's chromosomes don't all get together and all your dad's chromosomes don't all get together. They do independently assort. So that's called independent assortment. Does that make sense? And it doesn't. So that's another thing that happens in the first time around. Like we have crossing over happening in prophase 1, and independent assortment happening in metaphase 1. But crossing over does not occur in metaphase 2, and independent assortment does not occur in metaphase. No. Crossing over does not occur in prophase 2, and independent assortment does not occur in metaphase 2. Make sense?
Oh, can you say it one more time?
So crossing over, or independent assortment?
Independent assortment.
Alright, so you have one from your mom and one from your dad.
Yeah.
Right. We don't see, so if your dad's red,
Yeah.
okay, and your mom's blue, we don't see all the red ones on this side, right? We don't see all the blue ones on this side. We have like a mix match, like a mix and match. So you can take like one of your moms on like here, and then dad, mom, and for the other one's dad, mom. Like it doesn't matter like which chromosome is lined up where. It's just that they all act independently of each other. They don't all coordinate.
That makes sense.
Okay. Yeah. All right. So, what's your so? Any other questions? Yeah.
I have a question about like the, I guess the core or whatever. Okay, like how we have, so with mitosis, do we have only two spheres, or do we have like
No, we have kinetochores and mitosis as
Okay,
well.
okay.
Yeah, so this is the mitosis one. Kinetochores appear at the centromere. Right. So anytime you attach a microtubule to like a chromosome, you need a kinetochore. So it happens in mitosis and meiosis. Yeah. Good question though. Any other questions? All right. So what's the next phase here after metaphase 2? Anaphase 2. So what happens there? Separation of sister chromatids. Very good. All right, and then what else happens? That other step. Okay, your non-kinetochore microtubules lengthen. All right. And then let's bring them home. Telophase 2. What happens? What happens? So the nucleus, or the nuclear membrane, or envelope, whichever you prefer, reforms. What happens to your DNA? Yeah, your DNA loosens. And what happens with your microtubules? What? Well, yeah, they disappear altogether. Yeah. All right, and then we have that last step that's not in meiosis, but it's still part of the cycle. Cytokinesis, right? Cytokinesis. And what happens in cytokinesis? So you replicate your cytoplasm and cleavage furrow forms. The cleavage furrow forms also in this first cytokinesis. Right. Yeah.
How come at the end of cytokinesis the first time, you have 2 haploid cytoplasm and 2 diploid cells?
So at the end of this, you have 2. Okay, because what do you need to be a diploid cell? 2 sets of not DNA. What kind of chromosome? Really want to talk about? No, not quite. Homologous chromosome. Right. To be a diploid cell, you need both sets of homologous chromosomes. Right. You said that earlier. So a diploid cell is when you have both sets of homologous chromosomes. Right. So what happened there in anaphase 1? Yeah, we separated the homologous chromosomes and we put one into each cell. Now, each homologous chromosome has 2 what? Sister chromatids. So you still have the replication of each one, but we did separate them. So they become haploid at like after cytokinesis or after telophase 1. Does that make sense?
Yeah.
Why are we trying to?
I just have to like think about like 5 times until it sticks. So you're seeing it.
Alright, so here we lined up homologous chromosomes. So one from dad, one from mom.
Yeah.
That code for the same general material. And then we separate them, right? So we separate homologous chromosomes.
So like technically, or not technically, you still have 46 chromosomes. They're just not like 46 sister chromatids, but they're not.
So 92, right? We got it. But yeah, you're still at 92 sister chromatids, but they're not together anymore. Yeah. Until anaphase. Yeah. So we separate them, and then they're not in distinct cells until after phase 1.
All my plates ask for the but if you're here,
You have a question? What's the guy?
But
I feel like my hands are
like, so it's like, if you look at mitosis, it's kind of like you have like one pair, and you split them, it's still like a diploid, and it's like, yeah,
take it.
would you see like, would you see like 2 pairs, so it's like,
Oh.
like, you know, like, how,
Wait, what?
it's like you have
Like depending on how.
two pairs, and you're just splitting the pairs apart, instead of, like, in mitosis, where it's just one pair, and then it's like 8 apart.
That's what I got.
Like, it looks like it's.
We have another activity that we're kind of go over this. So if you're still confused after that activity, then we'll do it more. But let's see if that other part clears up some confusion that we have. Okay. So yeah, that's meiosis. Okay. So alright, we kind of have to hurry. Okay, so there are 3 different ways that we can go through. So we want to make a whole bunch of different types of cells because evolution really likes variation. So there's 3 different ways that we make variation here. All right, and it's independent assortment, crossing over, and random fertilization. So we saw independent assortment. What was that? So what stage was it? Let's start there. So metaphase what? So it's in metaphase 1 that so what lines up in metaphase 1? So homologous chromosomes line up, and they line up independently from each other. So they don't like look at their neighbor and be like, oh, are you from the mom or the dad? Oh, you're from the dad. I'm from the dad. Let's get together. They don't like do that. They just kind of focus on their own thing, and they're like, all right, I have to go line up on this metaphase plate. I don't give a fuck about anyone else. All right, we gotta go line up on this plate, and then we're gonna separate. All right. So they line up independently from each other. All right. And so because of that, like if you were to have all your genetic material from your mom on one side and all your genetic material from your dad on the other side and separate them and did that every time, you're gonna have like 2 of the same general cells, right? Like when you go through this, like all of your red ones would be over here and all of your purple or blue ones would be over here. And so when you come to the end of it, you'd have all red in these 2 cells and all blue in these 2 cells. Right, so all red here and then all blue here. And so because of that, it's like all the same. Like these 2 are the same. But if you do this independent assortment, these 2 can be different from each other, and these 2 can be different from each other. So it creates more variation. These things are more variable now. Does that make sense? So that's the thing that increases variation. All right. So crossing over also increases variation. Why does crossing over increase variation? Yeah, because you're randomly exchanging DNA with your homolog. So 2 homologs make a homologous parent. So you're with homologous pair. I'll just put that. So that makes sense, right? Because it's again that like if we all had the same DNA and we just separated, like randomly assorted, some could have some DNA and another one could have another. So you increase variation. And the last one is random fertilization. So that one is that you make all these different. So we make all these different haploid cells, right? And like if this is all an egg cell, we make like a whole bunch of different egg cells or a whole bunch of different sperm cells. So actually, it's more concerned with sperm, right? So if you make 4 different sperm cells, there's no selection. So that like biology doesn't say like, oh, this one's the best, so let's do that one, right? It just, you have each one of these has a complete 1 in 4 chance of fertilizing the egg. Okay, so there's no like selection at the fertilization level, so it's just random fertilization, right? So no selection. So you're not selecting any special properties of the sperm. So there's no selection at fertilization. Does everybody kind of see how all these increase variation within a population? Alright, cool. So what we're going to do next is we're going to do kind of like what we did last time with this chart, and we're going to keep up with the number of chromosomes and chromatids and DNA in a human. So we have your diploid of what? How many chromosomes? 46, yeah. All right, so let's go through this. So if we're in G1 phase, how many chromosomes do we have? 46. And how many chromatids do we have?
46.
So each one has 1 chromatid, so we have 46 chromatids. So this is like your reference spot, so we have 1 time, right? All right, so S phase, what do? We replicate it, so how many chromosomes are we going to have?
46.
So still 46,
I said right.
and then how many chromatids do we have? 92, okay? Because when we replicate our DNA, we put them on sister chromatids and attach it to the same chromosome. So it's still a singular chromosome, just separate chromatids. All right, so how much DNA do we have?
2.
So 2 times. All right, so we're going to go through G2. So how many chromosomes do we have? 46 chromatids, 92, right, and then times 2. So prophase 1, what happens? How many chromosomes do we have? 46 still. Number of chromatids? Still 92, and we're still 2 times. All right, metaphase 1, same thing, right? 46, 92 times 2. All right, because prophase 1 and metaphase 1, all we're doing is moving it around, right? We're not doing anything with the numbers. So anaphase 1, what happens?
It splits. It separates.
So it splits. So how many chromosomes do we have?
92. Oh, 23.
These things aren't connected.
Dang it.
So how many chromosomes do we have? We're not separating sister chromatids. The reason why we did it last time was because we separated sister chromatids and we made 2 distinct chromosomes. So in anaphase 1, we're not separating sister chromatids. So we're separating which aren't attached, right? So we still have 46 chromosomes. Does that make sense?
Yeah.
All right. So how many chromatids do we have?
92.
Still 92. All right, and we're still 2 times. So then we separate that to where so we go through telophase and cytokinesis. So at the end of that, how many chromosomes do we have in each cell?
That's 23.
So we have 23. Very good. All right, and then how many chromatids? 46. All right, because we're putting these chromosomes in distinct cells. So how much DNA do we have relative to G1?
Half.
Wait.
Twice.
No, not quite.
Same?
Same. We have 1 times, right?
Because it's the same amount of
Because
chromatids.
we duplicated it and then we took half and put it in 1 cell.
Things, but it's the same amount.
But yeah, it's a different type, right? We don't have both homologous pair, but we still have 1 times the chromosome because we only put it into 2 different cells. Because we had 2 times and we divided it into 2 different cells. So 2 divided by 2 is 1, right? All right. So prophase 2, what happens?
Same.
So same, 23, 46, 1 times. Metaphase 2, 23, 46 times 1. Anaphase 2.
It's half.
Oh, no, now you're splitting it, so now
So now we're separating sister chromatids.
This is half.
So this is
Oh, it's half.
46, right? because we're taking a chromosome that has two sister chromatids and we're splitting them into two distinct chromosomes, just like in metaphase 1. Mitosis and mitosis and meiosis 2 are essentially the same thing, just with different numbers of chromosomes. Right? They're very similar processes. So in phase 2, we have 46 chromosomes. How many chromatids do we have? 46. All right. And 1 time. So then we're going to divide these cells. So how many chromosomes do we have? 23, yeah. All right, and then we have number of chromatids is 23. That makes sense? So then how much DNA do we have? That's where we have 1/2. So what that means is we create how many cells? So we make 4 new cells with 23 chromosomes and 23 chromatids, and they all have half of the DNA. So that means we can take 1 from a mom and 1 from a dad, each have half, make it in half times 1. So if we fertilize it, we make it times 1. Does that make sense? Yeah. All right. So the next one we're going to do is we're going to label this, just to make sure we have our chromatids and homologous chromosome nomenclature all set. Okay? So, if we have this picture right here, this box is around what? Okay, so the black box means it's a diploid cell, right? So it's diploid. Because why is it diploid?
Because there's homologous pairs.
Yeah, because each chromosome has a homologous pair in that cell. So then, so if we label the sister chromatids and homologous chromosomes, so in this box, what do we have? So this box is this thing right here, right? So we have 4 sister chromatids and how many homologous chromosomes?
2.
So we have 2 homologous chromosomes. All right, so it's these 2 things right here. And where are the sister chromatids? All right. So where are they? So each one, so these 2 distinct things right here, so 1, 2, are sister chromatids, right? All right, so how many chromosomes do we have in this picture? No, in this whole thing right here. Oh, sorry, hold on. So in this whole thing, so how many do we have in this whole picture right here?
How many what?
How many chromosomes do we have?
46.
So we have 46 chromosomes. How many homologous pair? 23 homologous pair. And then how many chromatids?
What is that number?
96.
So 96.
46.
What's 46 times 2?
92.
It's 92. Right?
What did I say?
96? Yeah. Okay, so 92. I know it was like a whatever. So 92 chromatids. All right, so what cells can be in this stage right here? That we have 46 chromosomes, 23 homologous pair, and 92 chromatids. What? So somatic cells in what stage? So can we do it in G1? Can this be a G1 cell right here?
No,
Why not?
because it has one. Yeah,
So how do we know that it's replicated? Right, because each chromosome has 2 chromatids. So we know it can't be in G1. Can it be in S phase?
yes.
Yeah, so it can be in S phase. What about G2?
Yeah.
It could be in G2. What about prophase 1? Alright, so prophase 1. What about metaphase 1? So metaphase 1. What about anaphase?
What part of
Okay, yeah, so that one's kind of ambiguous, right? It's whether they're attached or not. So I'm going to say yes to metaphase 1. Right. To, I mean, yeah, to anaphase. But if they are separated, then it's not. Right? But these are, I mean, if it's together. All right, there, yes, okay. So anaphase 1. What about telophase 1? So why not telophase 1? Because, yeah, they become distinct cells after that. So we can't do it here. All right, so these are all of our phases that can have this kind of genetic setup. All right, so here we have...
Is anaphase before they're separated?
So at the beginning of anaphase, they're together, and at the end, they're separated. So it's kind of ambiguous for this picture, right? So it's probably a bad phase to look at in this context, but I'm going to say yes. But that's 50% arbitrary on my part. Right. You can make an argument for it not being. Okay, so here, right here, we have, so is this haploid or diploid right here? So this is haploid Y.
Because there's zero homologous pairs join one side.
Very good, yeah. Okay, so in this picture right here, where are the homologous chromosomes? There isn't one, right? Because there's only one of these things. All right, so if you're a haploid cell, you don't have any homologous chromosomes. So where are the sister chromatids? Right, the two individual things like we had before, right? Sister chromatids. All right, so how many chromosomes are in this picture right here?
Oh, that, I mean, 23.
So 23, right? So 23 chromosomes. How many homologous pair? Yeah, 0, because there are no homologous chromosomes in this picture. And then how many chromatids? So 46 chromatids. So what cells can be in this phase? All right. So
G1.
all right. So can G1 be in this phase?
No.
So why can't it be G1? Because there's no homologous pair. What about S phase? Well, why not in S phase?
There's no homologous
Again, because there's no homologous pair, right? In S phase, you'll have both homologous pair, and you replicate all of your DNA. Does that make sense?
S phase doesn't happen twice.
No, it doesn't. It only happens once, right? So S phase G2, can it be in G2?
Nope.
No, because no homologous pairs. Prophase 1, metaphase 1, can it be in those? No. Anaphase 1? No. Telophase 1?
Yeah.
So yes, right? Because, so it can be in telophase 1. Because after telophase 1, you have each cell having a, like that's where we put our separation of homologous pairs into each cell. Right? What about prophase 1? I mean prophase 2.
Yes.
Yes, right? So prophase 2. And then prophase and metaphase are, we said, were the same. So metaphase 2. What about anaphase 2? So this one's not as ambiguous. So why are you saying no here?
I need 46 chromosomes.
No, because the sister chromatids separate.
The sister chromatids in this picture are still attached to each other.
Yeah.
And in anaphase, we at the end of anaphase we separate the sister chromatids.
Couldn't that be the beginning of anaphase?
Yeah, it could be the beginning. I had like a disclaimer, right, that all the stuff we talked about at the end of it. At the beginning, yeah, it will look like this, right? Which is the end of metaphase 2. That's why we put metaphase 2. But once you go through anaphase, we separate the two sister chromatids. So we can't be in anaphase there. Alright. So let's go through with this. Is this cell right here haploid or diploid?
Oh, halfway. There's two.
So it's not haploid if there's two. It's diploid if there's two, right?
But they're not. Oh, I see.
Right, so this is right here is a zoomed in version here. So these things aren't attached like they were before, right? So these are distinct chromosomes.
Oh, so they don't have to be sister chromatids to be.
They don't have to be a homologous pair to be diploid.
They don't have to be. No, you just need one. So in this picture right here, we have two homologous pair. You have to, you have to have both homologous pair to be diploid. But here we had two homologous pair, each with two sister chromatids. Here we have two homologous pair, but how many sister chromatids?
You don't have
One
to have
sister
two
chromatid.
sister chromatids to be diploid.
Diploid. Yeah, you don't have to have two sister chromatids to be diploid. No. So here we have a diploid cell right here, because there are how many? So here, so here we have two homologous.
They don't have a sister.
Yeah, technically yes, you're right, but just for nomenclature's sake, makes it easier to talk about. So we have two homologous chromosomes here, right? And then how many sister chromatids?
0.
So 0 or 1, depending on how you look at it, right? So I'm going to say one sister chromatid.
How is that? 2 non-sister chromatids. Right?
Yeah, okay, we have each one of these. Yeah, so that's so 2 non-sister chromatids.
Oh, that's less of that. Okay.
Yeah, that's a good way of putting it. Does that make sense?
So like 2 potential sister chromatids.
Nah, sis.
So one potential sister chromatid.
You can say sister.
So this right here is, so this strand right
Something
here
like that.
is a potential sister, is one sister.
Potential. You can't be. There's no halfway. You gotta be in and out.
Yeah, give it a nod.
Philosophical.
Stepsisters.
Y'all see how this is 2 distinct chromatids? That's all I want y'all to recognize right here. Okay. You can write a paper on what you want to call it later. Okay. So we have diploid cell. So how many chromosomes do we have in this picture?
46.
So 46 chromosomes. Because these two right here are not sister chromatids. Right? So how many homologous pair do we have?
23.
So 23 homologous pair. And then how many chromatids do we have?
46.
So 46. Because these things are not replicated. So what are some potential phases that this can be in? Can it be in G1?
Yes.
Yes, right? Because this is just unreplicated, right? So G1. So can it be an S phase? No, because that's where we replicate it. Right? And we talked about how all these are what it's going to look like for all the way up until metaphase 2, right? Does that make sense? So this can only be in G1. The cell can only be in G1 phase.
That's it?
That's it.
I hope we get something like this.
Does that make sense?
Yeah.
Alright, so what about this picture right here? So are we haploid or diploid?
Haploid.
So we're haploid. Why are we haploid?
No pairs.
Because each thing is just, so there's no homologous pair, right? So haploid. So we don't, so how many, so right here, is this a, how many, where's our homologous pair? So it's non-existent, right? And then where's our sister chromatid? Right, so we only have one chromatid without any sisters, right? So you have one chromatid. All right. So how many chromosomes do we have here? So 23 chromosomes, and how many homologous pair? So 0 homologous pair, 23 chromosomes. Then, how many chromatids do we have? You got 23 chromatids. So what are some potential phases that we can be in? Can we be in G1?
You don't have no pay?
We don't have any homologous pair, right? So S phase. So yeah, that's all right. All the other ones look like this, right? So we're not gonna go through all of them. So where we left off was metaphase 2, right? So metaphase 2. So after metaphase, so right here. So this one's kind of, we get back to ambiguity. So here we don't have both sets, right? So we don't have both ones. So we can't really be in anaphase. We can't really be in anaphase 2. Does everyone see why we can't be in anaphase 2?
Yeah.
If we had another one like this, actually, this right here could be in anaphase 2, right? Because we separated each one, right? Anaphase 2. You'll see why this can be an anaphase 2? Because we separated the sister chromatids, but we still, yeah. Never really thought about it that way. All right, that's cool. All right, so anaphase 2. So can this be in telophase 2?
Yes.
Yes, right? Because that's when you separate your two things into two distinct cells. Telophase 2. And so after telophase 2, you have the setup like this. So does anybody have any questions on this page? Does that make things a little bit more sense? Or confuse things a little bit more? Either one. All right, very good. All right, what we're going to do is, if you want to stay after, we're going to go through this bottom picture, but we're just going to go through this and then start the multiple choice. Because we're running a little bit over. Okay. So we see some similarities and differences between mitosis and meiosis. So let's just go over similarities. So let's compare the two with these properties right here. So in DNA replication, how does DNA replication occur compared between mitosis and meiosis? It's the exact same, right? So you have replication in S phase and replication in S phase. Right. So both those are the same process for both things. All right, so number of divisions that we make. Is this the same or different? Different, because why? Yeah, it's once in mitosis and twice in meiosis, right? Because it's metaphase, so it's prophase, metaphase, anaphase all 1, and then prophase, metaphase, anaphase all 2, right? So it happens twice in meiosis. So synapsis of homologous chromosomes. In meiosis, it does not happen. Does not occur. But in meiosis, it happens in prophase 1, right? Happens in prophase 1. All right, so the number of daughter cells and genetic composition. So how many daughter cells do we have after mitosis? So 2 here. And what's the genetic? So are they haploid or diploid? So we got 2 diploid cells, right? But after meiosis, how many daughter cells do we have? 4. So we have 4 daughter cells. So 4 haploid daughter cells. All right, and so the role, so what do cells that undergo mitosis play? Like, what is their role in the body? So they're somatic cells, right? So they're cells of the body. Well, what about meiosis? What do those cells do?
They're gamete cells.
So yeah, they're gametes, right? Which are involved in reproduction. Very good. So y'all see kind of the similarities and differences between these two? Alright, so go ahead and start on your multiple choice. And number 6, that's not a multiple choice question. And do that for the next like 8-ish minutes. The questions aren't that bad. They're not as bad as last time. But try to get through this. Alright. If you have any questions, yes.
Are you posting on YouTube? Is that what you said?
What?
Are you posting like the session on YouTube?
Yeah.
What camera?
What?
Camera.
Mhm.
Oh.